Hello, everyone. Always those lights that bother us a little bit. Thanks for the introduction, and it's a pleasure to be here again in person. I'm Rogerio Vivaldi. I'm the CEO of Sigilon, physician by training, and working in rare disease for the last 30 years. In today's presentation, we'll make some forward-looking statements, so please take a look on our disclaimer page. The reason I joined Sigilon more than 4 years ago was that I believe harnessing the power of human cells and aiming to deliver functional cure is the way to treat a broad range of diseases, rare and prevalent. Sigilon was created in 2016, and from the seminal work of Bob Langer and Dan Anderson at MIT, when they discovered a fibromer, a conjugation of a small molecule and alginates to avoid immune reaction against an encapsulated cell. In 2018, we executed a strategic partnership with Eli Lilly for type 1 diabetes, and in December 2020, we did our IPO, raising $145 million. Our first program reached the clinic in only 3 years, an impressive accomplishment for a novel platform. We are applying all lessons learned from the hemophilia A program into the next programs with a very seasoned team, and I will show you in a second, through a constant platform optimization and a fiscal discipline in our cash runway into 2024. This is the platform that we have, and I showed this many times, so I'll not spend time now. We'll talk more about this in a second. Wanted to lead a team of people that are experts on doing this work. So we learned many things in our first clinical program. 3 patients have been dosed from 300 million cells to more than 1 billion cells. The product was implantable in the omentum, and also the product was retrieved, something that you don't hear as common in many of the new modalities. Can you retrieve your drug product? Can you turn off your system? So we did this in the 3rd patient. We also validate our GMP manufacturing process, and our extensive investigation in the last almost 9 months after the clinical hold concluded that there is no definitive link between our product and factor VIII inhibitors development. No other tolerability or safety issues were observed. There was evidence of sustained factor VIII activity for 20 weeks in one of the 3 patients. And during the retrieval procedure of the 3rd patient, we identified spheres with the pericapsular fibrotic overgrowth, and I will call this PFO during this presentation, but no organ or tissue fibrosis. We recently submitted the clinical hold letter to the FDA, and they agreed with our conclusions and agreed to anticipate the closing of this study by year end. I really want to thank the patients for their participation and their commitment with this innovative treatment for their condition. The way that Sigilon is then really pursuing drug discovery has been informed by our learnings from this clinical experience. With this, we are putting novel methods that will allow us to predict better the PFO risk, in addition to other important optimization that we put in our platform. So, for example, we now have cryopreserved drug substance instead of a fresh drug substance. We have implantation through an image-guided procedure with local anesthesia instead of a laparoscopy. And then we have strengthened the sphere in terms of integrity and stability. And also we have optimized it through the cells that I will be talking in a second. So something I heard this morning in the first panel, that how to test the allogeneic cell therapy in your preclinical studies. And that's been a challenge for everybody here, because you have preclinical, which is our xenogenic environment, but then you have to test your human cells. Okay, do you do non-human cells and test, Okay, this is not the final product. Do you test in xenozenic? Well, this is not the final environment. So again, we really have, and I want to spend a little bit on these prediction models about PFO that can be viewed as a hybrid of a foreign body response and a transplant reject response. The first step, of course, it is the foreign body response to the outside of these spheres, and then followed by this transplant rejection that goes into all the adaptive immune response that you all know, resulting in antibodies, IgM, IgG to the cellular proteins. So we have developed an assay that really goes into those three triggers here that are what we call the potential triggers to the PFO. One is what we call mediated by the foreign body response, and is mediated by a fibromer. The trigger two is the inflammatory response that is mediated by secretomes, which means what the cells are producing, cytokines or danger signals. And the trigger three is the response of the lymphocytes to the protein antigens that are released by the encapsulated cells. These include the product or HLA molecules. So what we have developed is this, an assay cascade to rapidly and systematically evaluate the PFO response to our encapsulated cell products. The macrophage attachment in your left measures the early step of the process. An overnight attachment of human macrophages, shown in red, would mean that this is a no-go because this product probably will cause PFO in the clinic. Then the humanized mouse model has a full human immune and human blood system. With that, the PFO reads out as the clumping of the spheres after implantation and accurately models clinic PFO to the encapsulated cell products. And the final preclinical models are non-human primate studies where allow us to test the long-term PK durability in the absence of PFO. So we are very excited, and just to give us a sense of size, this year we run more than 400 animals that have been humanized to test the different formulations that we have. So in December, we have prioritized our efforts in order to really execute a fiscal discipline with our cash, and we prioritize it in both diabetes and MPS I. So in diabetes, as I said, we have a partnership with Eli Lilly. We received $75 million up front, 415 milestones in addition to royalties. And there, our aim is to develop a cell therapy product that does not require immunosuppression and achieves independence from the administration of exogenous insulin. I can put a personal note here. I have type 1 diabetes myself for 49 years, so I'm looking forward to celebrate my 50th anniversary next year, and I can tell you that I dreamed for the last 20 years of something that could encapsulate islet cells. So to have a consistent supply of cell for patients, we are using stem cell-derived islets that contain the glucose-responsive insulin-secreting beta cells that are missing in type 1 patients. Induced pluripotent stem cells are differentiated to pancreatic islets with a multi-step protocol that uses our knowledge of embryonic pancreas development. We have been focused really on developing this protocol to be reproducible, that using scalable process that can be easily implemented for GMP manufacturing of the product for the clinic. Our product generates stem cells islets that are similar to the human cadaveric islets with a high percentage of beta cells, high levels of insulin content, and high levels of glucose-regulated insulin secretion. I'm very happy for the first time to show some numbers with you that in our last recent in vitro and studies, we have accomplished more than 60% of beta cells that express C-peptide but not glucagon, and the nuclear beta cell transcription factor, NKX6.1. So we are extremely excited with those baseline, what this means in vivo. So again, to protect the islets from the immune system, we are encapsulating them in our spheres to generate our diabetes drug product we call SIG002 for administration to patients. We have shown that 002 is efficacious in the streptozine-induced diabetes model, and as shown here, 10 weeks in vivo after one implantation, 002 in green and purple, you can effectively reduce the blood glucose levels. And here we are showing an oral glucose tolerance test, and also the overnight fast that we have for the glucose. So it's a picture that, one, does not different than the non-diabetic animals that are in these studies. We are very excited with this. This is day 70, and want this to repeat even further and further. Shifting gears to talk about the MPS I. I have been involved with lysosomal disorders for many years, but we wanted to take the best shot. So what does this mean? As we previously communicated, we wanted to really work on the product candidates that could address all the manifestation of these patients, including neurologic manifestations, using transporter molecules designed to penetrate the blood-brain barrier, and molecules designed to extend the plasma half-life, as the enzymes are very short half-life. We believe that makes sense to pivot this right now. So we are expanding the program for MPS beyond what we had initially that would not cover into the CNS. Of course, with this, we'll not move in 005 into clinic next year, but our timelines and costs will be pushed back, but I believe that is the right thing to do for patients and for ourselves. We'll have the best shot and we'll have the best product. This offers a potential for sustained substrate reduction versus what you can see here, the peaks and troughs. You can see lightly the peaks and troughs of an enzyme replacement injection. Here, we just show one of the 6 months studies we conducted with this drug product, comparing with the enzyme replacement therapy arm. And as you can see, the bone correction, which is something very difficult in lysosomal storage disease, has happened with the arm that has received the implantation of the cells producing IDUA versus the enzyme that was given to them in clinical doses. But more exciting is that once we apply all these molecules to extend the half-life and to cross the blood-brain barrier, we reduce dramatically the substrate in all tissues, and we also reduce in the brain by more than 32%. So with this, I think that we can really bring to something that will help patients in a big way. We have finally successfully developed tech-transferred manufactured GMP drug products, which we were using in our phase 1, and we are now continuing our efforts on manufacturing improvement to meet the needs, which means scaling up the cell, automation in the encapsulation so that we can have a real off-the-shelf product. Our vision is really to become a cell therapy powerhouse, leveraging the technology that we have across multiple cell types, because it's important to mix many cell types, targeting a broad range of important indications with the potential to bring transformative new treatments to these patients. With that, I wanted to thank you all for your attention.